Log on, tune in, find out. Another good idea from Cambridge.
And I hope we'll stimulate some interesting questions. Daniel.
Yeah, thank you very much. Thank you to the audience, the organisers, for inviting me to give this talk. And I was sitting here. Sorry?
I don't think it's on.
Yeah.
I think it's on. Okay, I was thinking, how does this fit into a conference at a math research institute? And I guess I realized, I remembered the title is New Data, so at least it fits under New Data. And this is an overview of what we're talking about. And if there's enough time, I might say a little bit about something completely different. Might talk about phylogenetic networks for a few minutes, depending on how this goes. Because I know there's a lot of people here who are interested in networks, and so I'd like to share what we've been doing just recently, if there's time. Okay, so genomics, just let me remind you where it all started, here in the kitchen of Tübingen Castle, where in 1869 Friedrich Miescher discovered DNA. So he was looking at bandages from wounded soldiers, and he found these stringy molecules, and he called them nucleins. So it all goes back to Tübingen. And just to remind you that genomics, of course, is the study of the genome sequences of individual organisms. And the basic things that we do in genomics is we like to sequence and assemble stuff, annotate and analyze genomes, compare genomes, and so on. And you know that due to advances in sequencing technology, the number of projects in which people are trying to assemble genomes is growing at a very fast rate over the last 10 years or so. And most projects focus on bacteria because they're smaller and there's more of them, different types, but eukaryotes and so on are also of interest. And you can see in this plot here also there's more and more metagenome projects that people are working on. And I want to say something about the GBA project, which is not a metagenome project. It's a project where it's a joint project between the JGI and the Deutsche Sammlung für Mikroben und Zellkulturen. So these guys in Germany, they got all these different types of bacteria and so on, and so the people at JGI are sequencing these, and the idea here is to try and get a nice spread over the different phyla of bacteria, because we want to use databases of known sequences as a reference database to analyze environmental samples. And the problem is that the database that we have at present were not constructed with that in mind. They're biased towards very few organisms that are of some interest to somebody, and they were not. The NR database was not designed as a taxonomical reference database. So in this project, they're looking at sequencing microbes of underrepresented clades, and one of the interesting things in their paper is this plot here. And it's interesting for this conference also because phylogenetic diversity is used here in a useful way. And so here you see on this axis 40,000 sequences taken from the Green Genes database. These are 60S sequences, and most of those are from environmental samples. So these are somebody's grabbed a 60S from somewhere. We don't really know what the species is, but we've got the sequence. And if you measure the phylogenetic diversity of the whole database, you get about 1,000 units. And I'll just remind you what phylogenetic diversity is in a minute. And so this is this grey thing up here. So if you look at all the sequences, now if we restrict our attention to those sequences for which we know which organism they come from, we're already down to less than 200 units of phylogenetic diversity. Now if we restrict our attention to those sequences for which we have a fully sequenced genome, then we're down to less than 20 units of phylogenetic diversity. So what this is telling us is there's a huge discrepancy between the sequences that we have in databases and what we believe is out there in terms of phylogenetic diversity. So many approaches in metagenomics, you end up comparing sequences that you got from the environment against sequences that are in in fully sequenced genomes, and you can see that that's really not telling you much about what's out there. Yeah, just to remind you, phylogenetic diversity has probably been already defined a number of ways here. So you have a big tree and you have a subset, and how much of the big tree does the subset cover? Okay, sequencing, I don't have to mention that sequencing is getting crazier and crazier. The latest Alexa sequencer, the Illumina HiSeq 2000 with the new kit, whatever, produces 300 gigabases of sequence in one run. So nobody knows what to do with these sequences, and some people recommend just subsample. So if you do all the sequencing, just use a subsample of the sequences, and then you're fine. And now here's an interesting slide in this context given to me from by Volker Meyer. And here the question is, given $30,000, how much sequence can you get? If you use a 454, for $30,000 you roughly get 1 gigabase per sequence. This was true at some point in time. Of course, everything's shifting every day. The numbers have changed. But at some point in time, you could get, for $30,000, you get 1 gigabase of sequence. And to blast that against the NR database using BlastX, that's going to cost you $7,000 in CPU money if you do it on the Amazon cloud for 50 cents per hour. Now, you spend $30,000 on the old style Illumina, you'd get 60 gigabases, and now you already have to spend $240,000 to do the blasting. And now the next generation thing here, for $30,000 you now get 196 gigabases sequence, and so you're looking at $600,000 just to blast your sequences against the NR database if you're doing some kind of metagenomics thing. So the good news for people who are interested in bioinformatics is that bioinformatics is now where the money should be going, and not to the labs. Okay, what is metagenomics? I think this name was invented to describe the study of DNA of uncultured organisms. We all know that this is what biologists like to do: put some bacteria that they're studying into a dish, feed it, and then study its properties. But most bacteria don't go along with that. So people don't know. Some require some other bacteria to be present, and so on. If they're not there, their bodies are not there. They just die, and so on. And so people will want to study these bacteria anyway, so we do it by sequencing their DNA. And if we take a genome just to be the complete DNA of an individual species or organism, then a metagenome is just, I shouldn't use the word community, just assemblage of organisms here. So you've got a handful of all the DNA that's in there. Okay, people don't like it when you use the word community because it sounds like the bacteria have something to do with each other, and that's obviously not always the case. So assemblage of organisms, I should change that slide. And then, the metagenomics, you've got all the DNA, then metatranscriptomics, you've got all the transcripts that are present at some particular time, and metaproteomics, if you can capture all the proteins, and so on, it goes on and on. There's many different projects in metagenomics, and everyone's studying all kinds of environments, the soil, medical samples. There's a human microbiome project. There's other projects that I'll mention briefly later. And just as an example, for those of you who are not familiar with metagenomics, one of the big projects of the last 5 years or so is the Global Ocean Sampling Expedition, where Craig Venter and a bunch of people from the Venter Institute set off on a round the world sailing trip, more like an extended holiday, but along their way they sampled water. I think they collected 200 liters per sample and then filtered it down. Then they size filtered everything that's in the sample. They didn't want viruses, they didn't want things that are too small, they didn't want things that are too big, and then they did a bunch of Sanger sequencing. And two of the main results in the special edition of PLOS Biology that came out in 2007 is one, that they were surprised to find a low abundance of clonal organisms, which is explained by this plot here. If you think of this as the genomic axis of a particular Pelagibacter organism, and then this here reads taken from the different samples that have been mapped against it, and the y-axis is percent identity. So you can see that you've got the spectrum from 100% sequence identity all the way down to 50%. So there's a huge so it's not like every organism of this type has exactly the same genome, but any level of similarity that you're interested in, you'll find some organism that happens to be 65% identical and so on. Obviously, this is probably due to the fact that they were taking hundreds of liters of water and taking stuff out of that. But that's an interesting find. And the colors here represent the different samples. You can see there is some samples, this green sample, whichever one that was, is more similar to that than some of the others. So that was one result. The other result was that if you look at the protein sequences that you can identify in their reads, they came up with 6 million, I guess, new proteins. And if you plot a rate of discovery curve, then they were still in a linear path. So if they'd done twice as much sequencing, they would find another 6 million proteins that no one's seen before. So that was also quite interesting. And that's one of the reasons why they did Sanger sequencing in the first place. I guess, nobody can really afford to do Sanger sequencing these days, but if you get average read length of 800 base pairs, and that's long enough to capture open reading frames from genes. If you're just doing miniature solid sequences of 50 base pairs, you're not going to be able to come up with these kinds of numbers. Here's another example of what people like to do. They like to take mice here, a beast mouse and a normal mouse, and then try and correlate what you see in the gut microbiome with what you see phenotypically. So is there anything different between an obese mouse and a regular mouse if you look at what's living in the gut? And then in this paper with, I think, 4 data points, they proved that there is a significant difference in what bacteria you can find and also what kind of genes you can find. So I think at the moment, for a while in metagenomics, you could, so this is one example of the kind of studies that people are doing. We're doing some metagenomics in Tübingen where we have a mouse model where the mouse is a sterile mouse, has no gut flora whatsoever, and then we can give it particular bacteria. So here you can have some E. coli and some of this and some of that, and then we're going to study the gut genome and see in a more controlled way what correlations we can find between sick mice and healthy mice and so on, and mice on certain diets. Okay, this is where I got into metagenomics in 2006 when we were together with Stefan Schuster at Penn State University. We were looking at the mammoth genome, trying to sequence mammoth DNA out of a bone that was found in the permafrost. And whenever you have ancient bones and stuff like that, you're kidding yourself if you think there's only going to be DNA of the host in there. Obviously there's going to be all kinds of bacteria and other things that have invaded the bone in there. And so we were interested in trying to get a handle on what kind of organisms we think are in that mammoth bone. And people are trying to do metatranscriptomics, capturing RNAs and then reverse transcribing them. And then in this small study here that I was also involved in, what happens if you go about it naively? You capture mainly rRNAs because that's the most abundant type of RNA in cells, and then but you can use those to do a taxonomic analysis, and then you get some messenger RNAs, and that'll give you some idea of what kind of genes are being expressed. But there's a lot more of that kind of stuff going on these days. And there was a recent paper where I think 600 gigabases of sequence were captured from 124 European individuals, and the goal here was to try and find a baseline gut genome for humans. And so this is a lot of data, and I was happy when I read this paper. Into the very last line they say something like, we use Megan, the program that we've been working on, to analyze the content. Okay, so that's just some of the types of projects people are looking at. You can what are the computational questions? So you start with an environmental sample, and then you do some sequencing, and then you do some kind of computation. So you get 10 million sequences, spend 10,000 hours doing some blast comparisons or running some taxonomic classifier or something. And then the 3 basic questions are, you want to get an idea of what kind of organisms are present. So it's the so-called who is out there question, and you've got the what are they doing question. You're trying to get a handle on what genes are present in the genomes. That's called the coding potential. Or if you're looking at metatranscriptomics, what genes are being expressed. And then the third question is, you want to compare different samples with each other, and in particular you want to correlate what you see in these different samples with some external parameters. Is there a difference between the lean mice and obese mice if you have lots of these mice, and so on. Okay, and there's all kinds of other questions. I'm going to be looking at blast-based approaches, but people are very interested in trying to use different machine learning techniques to cluster sequences. People are trying to assemble metagenomes, but we see that's not always going to be easy. If you think back to the that marine data where there's this widespread of different levels of sequence similarity, how are you going to know when one read still belongs to that genome or whether it's coming from somewhere else if you can't have a nice clean cutoff of 98% identity or something? Yeah, there's all kinds of things. So who is out there? What are they doing and how different do they compare? And these things, you keep in mind that usually when people are asking these questions, they're not just asking them just to answer these, but they actually have something else in mind. In medical, with the obese mouse and so on is pretty clear. Maybe you can come up with some therapy. You know, you change the gut genome of somebody and they suddenly lose a lot of weight or something like that. So who is out there? So one popular approach is people use targeted sequencing. They focus on some gene like the 16S rRNA sequence, and then they use a reference genome, reference phylogeny, and then they take these sequences from their environmental sample and try and place them into the phylogeny. And if they land up near a bunch of alpha proteobacteria, and they say, okay, this read probably came from alpha proteobacteria, and so on. And what we're more interested in my group is looking at random, proper metagenomic sequencing, where you randomly sequence DNA and then you try and bin this DNA taxonomically. And the way that we do it is just use the NCBI taxonomy. And you've heard, yesterday you heard about this kind of approach already, but I'll just briefly go over it nevertheless again. So you've got your different DNA reads, and then you compare them against a reference database, and then, okay, let's just skip those alternative approaches, and then you map them onto the NCBI taxonomy. So you have your read, you compare it against the NCBI NR database of protein sequences, then you see a number of reads, and you look at the number of matches, and you look at whether things match, and then you place the reads somewhere in the NCBI taxonomy, and I'll be discussing how this is done in Megan, the program that we've developed. So in more detail, here is your sample. You've done your sequencing. Now you do a sequence comparison against different databases. If it's a host-associated metagenome, like you're looking at a mouse gut, then you might want to compare against a mouse genome just to catch any sequences that come from mouse rather than from some bacteria. Otherwise, you look at something like the NR database. And it's similar for other types of projects. And what people usually use is something like BLASTX against the NCBI NR database. So you're comparing DNA reads against protein sequences. You might be asking yourself, so why don't we just do DNA versus DNA? And the answer to that is, if you can do that, you can take the NT database, which is the DNA database, or whole genome databases, and you can blast your reads against DNA sequences, but you will get less than 1% matches. Most sequences don't match anything, and it's not surprising if you think about that GBA plot at the beginning. We're not really trying to identify sequences for which we already know the genomes. All we can hope for is that we use homology to find more or less distantly related reference sequences. So you have to use BLASTX, and that's why all the hope that people have that just use those short read aligners. So people have spent a lot of time developing algorithms that can really quickly compare sequences against DNA sequences against highly similar DNA sequences, but they don't apply here, fortunately. So you identify a bunch of homologous, or hopefully homologous reference genes, and you get these BLAST. Everyone's probably seen these. If you ever use BLAST, this is what BLAST gives you, such records of significant matches. And then you can have a look in here and say, okay, this is the organism that this reference sequence came from, and then you can try and use that to place your reads. So usually you get more than one match. So say we have a read that matches these 3 different bacteria here. Let's see what they are again. Campylobacter, a Helicobacter, and a Volunella. Then the simple-minded approach, and I think you heard yesterday how to do this in a more intelligent fashion, is just go to the lowest common ancestor in the NCBI taxonomy, and then place the read here, and keep in mind that most likely it might well be that this is a new species. It's neither of these 3, but hopefully someday will be classified in this particular order here. And so this is basically a gene content approach. You have your read, you map it to a gene, and you ask which organisms contain this gene, and then you go to the lowest common ancestor of those organisms. And we like it because it's simple to implement. it's quite robust against false positives. People always worry about horizontal gene transfer. Now, if there's an instance of horizontal gene transfer and both the donor and the acceptor organisms are present in the database, then what's going to happen? The read's going to go to the lowest common ancestor of those two guys. So it's not going to be placed either in the one or the other. Obviously, there's going to be many cases of horizontal gene transfer that we don't see in the database, and then it's not so easy. Okay, here's just a small detail. So one of the things that we used to do is, what happens if only one organism or one read hits a particular organism in the NCBI taxonomy? Then we used to throw that away. Say, okay, that's not significant. So say we have this scattering of reads on a particular part of the NCBI taxonomy, and say we are only going to believe that an organism is present if it gets at least 50 reads. Seems like a good enough number. Well, we used to just throw all these reads away because they're not significant. Now we just push them up the taxonomy until you have a node that has enough reads on it. I mean, it's not rocket science, but it's implemented. There's lots of stuff that's really cool, but it never gets implemented. Okay, you heard about the NCBI taxonomy yesterday. Last time I looked closely, it was 670,000 nodes in that taxonomy. I'm sure it's way over 700,000 now. It's growing at an alarming rate. When I started working on metagenomics, it was at 280,000 nodes. And so that the taxonomy is getting bigger and bigger. And remember, it's organized in this way: kingdoms, phylums, classes, orders, families, genus, and species, and there's a couple of other unofficial levels of taxonomy in between. And in our program, when we try and represent results, we always draw it from left to right. So you get a picture like this in Megan. There's the root of the NCBI taxonomy, and then here's the first level of the taxonomy, and the second level. And then Megan, you can unclaps and collapse at a specific rank, or you can click on individual nodes and say unclap this node, unclaps this node all the way down to the leaves, and so on. And so this is what a user sees when they open up our software. Give it a blast file. They see an overview of the analysis. Here we're looking at 50,000 reads taken from one of those black smokers, 3K under the Arctic Ocean, where there's this lady in Bergen, Ina Steen, she's got this little submarine that goes out there and picks up a little bit and brings it back to the surface. So that's quite nice. And this is one organism that she's particularly interested in, Desulfobacterium. And so you can see some reads have been assigned to it, but there's also, any of the reads along this path to the root might also come from that organism. It's just that they're not specific enough for us to be able to assign them with confidence to this node. So if we go over here, say, and have a look in more detail what's assigned to the Proteobacteria node, then here's one particular read, and it has these significant matches against these different organisms. So in this case, the program just says, I'll put you on that node because I can't decide where you come from. So this is a very simple-minded approach where each read is treated as if it's as an isolated piece of evidence. We don't actually make any use of the fact that there's all these other reads that they also have to go somewhere, and it all has to make sense. And so there's a lot of room for improvement in this kind of algorithm. Okay, the software here, you can search for stuff if you know what it's called. You can go in there, you can click on a node and say, show me all the reads that have been assigned to it. Then for a given read, you can say, show me all the matches that it has. And for a given match, you can drill down all the way down to the blast alignment. So one of the things, talking about new data, we have more and more of these sequences. You have, say, 100 million reads, and you want to be able to navigate that data. So you need to structure it, but you need to be able to drill all the way down to the individual blast matches. So you have to slice and dice the data in a nice way. And so using the NCBI taxonomy is one way to organize the data. What was that? I missed that one. Oh yeah, I already said you can collapse and uncollapse parts of the tree. So and you can do click on some nodes and produce some pie charts and stuff like that, summarize the data at different levels of the taxonomy. You can capture data, you can say, give me all the reads that have been assigned to a particular part of the NCBI taxonomy if you want to try and assemble those reads, do a CG content analysis and so on. Okay, so this is not very mathematical at the moment. This is more like software engineering. There will be a little bit more interesting stuff slightly later, mathematics-wise. Okay, so that's the basic simple approach to trying to classify reads by taxonomy. Now, what about the what are they doing question? So it's the same computation. We blast the reads against the NR database using BLASTX. So for each read we get a bunch of protein sequences, and now we want to navigate those protein sequences. And so we need something that's similar to the NCBI taxonomy, except that it doesn't classify by taxa, but by phylogeny, but rather by function. And so one way to go is to use what's called the seed. It's a classification of genes by so-called functional roles in subsystems. So a subsystem consists of a number of functional roles, and each genes are mapped onto individual functional roles. And the functional role can occur in more than one subsystem. And there's two servers make heavy use of this, the RAST and MG-RAST. RAST stands for Rapid Annotation Using Subsystem Technology, and MG stands for Metagenomics. If you think of this as a graph, it's roughly 13,000 nodes from the root through different levels of classification down to the individual functional roles. This is an example of a subsystem. We don't have to understand what all these arrows and that mean. All we know, for us, it's interesting that this thing has a name and it has a list of parts, the different functional roles, and so the reads will be mapped onto these different functional roles. And in Megan, if you look at data set here, I have a different data set. I'm not sure what it is. 136,000 reads roughly, and this is what you get if you click on the seed viewer. You get a high-level listing of all the different types of subsystems of present, carbohydrates, photosynthesis, and so on. And then you can click on each, and there's always a large number of unassigned reads or ones with no hits. And actually, for most reads in the NR database, we know what organism they came from. But for most reads in the NR database, we know which organism they came from. But quite often, we don't know what function they have. So you always have a higher number of not assigned when you're doing functional analysis. Then let's zoom in on protein metabolism. You can click through 3 or 4 levels of hierarchy until you get down to the individual functional roles here. So these are all functional roles, have something to do with GTPases, for example. And then you can click on stuff and say, okay, give me the breakdown of these particular types of functions. You can capture reads again by different assignment. I want to capture all the reads that have to do with GPTases and so on. And then you can go in there and have look at the individual blast matches and so on. And okay, so not that many people are familiar with SEED, but I think a lot more people have heard of KEGG, the Kyoto Encyclopedia of Genomes and Genes. And so another way to do functional analysis is to map your reads against the KEGG database, and here's the top level of the hierarchy of different KEGG pathways. So KEGG database has pathways, and each pathway contains multiple enzymes, and so here the reads are mapped onto enzymes, and then the enzymes appear in pathways. So here again you can unfold this hierarchical classification, drill all the way down, and at the very bottom you will land on a visualization of the actual pathway, and then the different enzymes that were involved are highlighted, telling you how many reads have been assigned to that particular enzyme in that pathway. And then you can do all the stuff that you can do with everything else. You can click on this guy and say, give me all the reads, show me the reads that have been assigned to this enzyme, and so on. Here's a different pathway. So what I like about this is you actually get some nice pictures from those guys. So here you can get an overview picture. So this is supposed to show all the pathways in one unified picture, and then the ones that are present in your data set light up. So we're only, I'm not saying that Megan solves the problem of doing a functional analysis of a metagenome. By no means. I mean, this is just a very naive first step in that direction. There's all kinds of interesting questions that are going to arise. For a start, if you have a pathway and you see some enzymes light up, does it really mean that that pathway is present in the data set or not? That's unclear. And what happens if, because just because it's green doesn't mean that all this enzyme, I mean, let me put it this way, there's all the questions that have to do with the following. Is this pathway, are all these green things that light up, do they all come from one type of organism, or is it 55 different ones? If it's 55 different ones, we probably don't believe that this pathway is going to be present, because it's unimaginable that 55 different types of bacteria have to get together to get this pathway going. But if there's only 2, maybe there might be pathways where, okay, when the 2 right organisms come together, they can supply the needs, the things that are necessary. So analyzing pathways, given that your data comes from multiple sources, might be an interesting new field to look at. And so the way this is done is obviously at the end of the day, you just look at your blast matches, you grab some identifier, then you have some big lookup table, and that tells you, okay, if I see this particular RefSeq identifier, then I know where that maps, which functional role that maps to in the seed database. And similar with KEGG, if I see a particular RefSeq, then I have a big table that tells me where all these RefSeqs go, and then I can assign it. So there's nothing particularly difficult about that. How do different samples compare? This might be more mathematical. Got phylogenetic networks in here somewhere. Okay, so this is a typical picture in Megan. If you load multiple data sets, and then you get the same taxonomic view, but you just get like little bar charts showing how the different data sets all compare here. And up here, here's a legend for the different data sets. So these are all marine data sets taken at different times. Some of the first 4 are DNA, and the other 4 are cDNA, so the second 4 are transcriptomic data. And here's the seed-based view. So here I'm just telling you that you can do this visual comparison, and you can zoom in, and okay, and then you can make one of these views, say how do they differ on a particular gene, a particular type of, how do they break down for protein biosynthesis? Can we see something interesting there? You can do the same with KEGG. You can, okay, and these pictures, and instead of having one color there, you have a little colored bar chart that tells you how the different data sets map onto those different enzymes. Okay, but okay, now this is where the phylogenetic networks come in. So what people also want to do is they want to, compare these using more sophisticated comparisons. So what you want to do is you uncollapse the taxonomy to a certain rank that you're interested in, and then you select those taxa that you're interested in. And then once I've made a selection like this, what I've basically got is I've vectorized my data. I've got a vector of length, I don't know, 25 maybe. So I've got 8 vectors, each of length 25, and now I can do I can move to the world of ecology. It's like somebody's gone out there, counted how many butterflies, birds, and so on, trees are in a particular habitat. Then you can use all these different measures that people have come up with. The good old distance, and so we've implemented these, and so you can say exactly what numbers you want to base your analysis on, and then you can just fire up this window, and you get a neighbor net showing how the different data sets might relate to each other based on that data that you've selected, and based on the ecological distance index that you've selected. Or you can do one of those multi-dimensional scaling plots, and you can do the same for functions. You can go in there and say, okay, I want to do an analysis based on seed. These are the functions that I'm interested in. Make a big data matrix out of that, and then run it through NeighborNet. And then you can look at how the taxonomical comparison matches with the functional comparison. But at this stage, it's all very experimental. So basically, I would assume what a biologist does is they click through the different options of computing a distance until they get something that looks like what they're hoping to get. So that's always a danger of this kind of approach. And here's another picture. So we haven't implemented yet, but we're implementing obviously something that's trivial from a mathematical point of view, but very useful in applications. Is just a way to map from metadata. You have some attributes assigned to each of your data sets. You want to color them in by some certain attribute, and then hope to see patterns emerging in the network. Okay. And just recently, so we had the networks in for a while, then I decided, okay, I'll put a tree in there as well, just because I know that not everybody is happy with networks. So here's something. So this is one project I'm involved in, looking at this kind of data. I'm also involved in looking at data from water treatment plants in Singapore, and so you can envision comparing two different completely different types of data in this way. 16S, so integrative analysis was in the title of the talk, and what does integrative mean? It really means we're trying to make a tool that people can bring all kinds of data, and so you can bring in the random metagenome data, but you can also bring in 16S data, transcriptomics data, proteomics data, and have them all in one unified view, so you can actually see everything through one pair of glasses. Okay, so this is a 16S analysis. Okay, then different ways to look at stuff. Okay, the outlook. That's good. So I can spend a couple of minutes talking about phylogenetic networks. Okay, so there is a number of large projects, and each of these projects, including this new project in Singapore, producing terabytes of sequencing data. So okay, terabytes, that sounds pretty bad. But just last week there was a conference in China. The Earth Microbiome Project met there, and they're looking at producing hundreds of thousands of samples, estimating 2 petabytes. They're estimating going to collect 2 petabytes of sequence, and if you think about all the data that each base for every base of sequence, you've got like so many thousand bases of bytes of annotation and so on. So we're talking exabytes of data here. So just to let me remind you, this is mega, giga, tera, peta, exa. So we're looking at exabytes of data. And so the guys who are leading this EMP project, guys from Argonne National Labs, they don't think this is large data sets. They say, so in other fields, exabytes of data are considered relatively small. So I don't see how we're going to deal with that. Okay, and there's all kinds of computational challenges. Obviously, data access, if you have those large amounts of data. So something like Megan can only take you so far in terms of being able to keep an overview over your data. Analysis methods have to get faster. One of the things is that I first said that each read is analyzed individually, at least in our approach, but it's also true that each data set is analyzed as if it's the only data set that ever existed. And what we really need is some giant machine learning algorithm that has this big model of all the metagenomes data that's ever been produced, and as more data gets produced, it should all start to fill in gaps, and things should come together in this huge one big model of all the data. So how to learn across multiple data sets. And this is something that, from a practical point of view, people want to correlate environmental parameters with what they see in a taxonomic or functional analysis. Okay, and this is probably the main thing here, if anything. If we only know this much about all that diversity, then we're kidding ourselves if we think very soon we'll have a deep understanding of what's in the metagenome. These are all the people involved with this work, and because I've got some few minutes, I'm going to move on to this. So I know it's not usually done that somebody gives 2 talks in one time slot, but this is just, so Simona and Celine are both here, and so in the last months we've been working on rooted phylogenetic networks. And one of the things that we just released last week is Dendroscope 3. So I don't know how many of you know Dendroscope. It started out as a tree viewer for large trees, and now it's moved on to hopefully become a platform for delivering algorithms for rooted phylogenetic networks. So I think there is quite a bit of interest in networks out there, but I think people will start using networks more the more software there is that they can easily use to produce such networks. And so some of the things that are in Dendroscope 3 are different consensus networks. There's some stuff that Vince and Kathy and others worked on, multi-label trees, take a multi-label tree, make it into a single label network, so-called software networks, hardware networks. We've been looking at this quite heavily just recently, hybridization networks, and we've been looking at tanglegrams, something that's been discussed here in the past before. And so one of okay, it's always bad to talk about your own software, but the thing I like best about Dendroscope 3 is it starts out like this. You load a network or a tree, and then you can just say, okay, my file's got more than one tree or network in it, so you can just zoom in and out. You can see more or less. You can Right? You can have a 100 by 100 grid if your screen's big enough. And the nice thing is anything that you then do applies to everything that's either visible if you haven't selected anything, or if you've selected some of the trees, it applies to all the trees or networks that you've selected. So I think this should make it easier to, when you're doing a multi-tree, multi-gene study, you've got 7 trees and you want to see them together and do stuff to them together. You want to reroute them in a certain way and so on. So dendroscope makes that easy. Tangograms is, I think Celine talked about this. You can click on 2 things here and say, just give me a tangogram. Then you get a tangogram of the 2 networks. And so we try to make the algorithm so general that it can be bifurcating, multiplicating. They don't even have to have the 2 things that you're comparing, don't have to have the same taxa. So we try to make it robust so that your biologist can just throw in some trees and see a tangogram without having to worry about, oh, it's not bifurcating. Oh, there's some taxa missing in the one. Or, oh, it's not a tree, it's a network. So that seems to work. And so the minimum hybridization network. So 2007, in the Newton phylogenetics program, there was some discussion about computing the hybrid number. We discussed Magnus Bordwich and Charles Semple was another were here, and they had come up with an algorithm for computing the hybrid number between 2 bifurcating trees. And then there was some effort in New Zealand to produce an algorithm for computing all networks, minimal networks for 2 bifurcating trees. So the problem here is you've got given 2 trees, you want to find a network with a routed network with a minimum number of reticulations that contains both trees. That's a hard problem. Fixed parameter tractable, if you know the number of, in the number of reticulation nodes. And so together with the student Benjamin Albrecht, Celine and I worked on this, and we came up with a nice algorithm that produces all possible networks in some sense that contain the 2 trees. And it's a very fast algorithm given that it's a hard problem. So it seems to work well even when there's like 19 reticulations or something in the thing. But the thing about that is, and we heard somebody say this morning, assume we have 2 bifurcating trees. Okay, we can assume that when we're in a mathematics setting, but no biologist has 2 bifurcating trees. Nobody ever, because the first thing you do is when you come up with the tree is you start contracting down the stuff that is dodgy, because why resolve something if you know you can't resolve it? And most algorithms that assume that you have 2 bifurcating trees make use of the fact. Now, in this kind of question, you're given 2 trees and you want to reconcile them using a minimum number of reticulations. It really hurts if some of these bifurcations are just randomly resolved, because you're going to spend a lot of reticulations trying to make up for the fact that, but it's not clear what really happened here. So I think it's important to have an algorithm that gets away from that. So we now have an algorithm that takes 2 multiplicating trees, and not that these are multiplicating here, but it doesn't make any use of binary whatsoever, and it can take 2 trees and compute a minimum network. Isn't it? And for those of you who are in the know of the technical stuff, it's not based on maths, so which is quite nice if you don't like maths. And okay, so we're taking the same data as for the binary. So we have 2 algorithms, one for binary trees and one for multiplicating. And then so we're trying to, obviously, to be sure that these algorithms are correct and so on, we want them to produce the same number of trees on the same number of networks on 2 bifurcating trees. It turns out that one algorithm produces 9 and the other one produces 12. So I asked Simona to look at these, and Paul Simona, who's trying to look at, try and compare 9 here with 12 here of such networks. It's not very easy. So 2007, there was a talk here, the tree of trees, where somebody was saying, let's use a tree to cluster trees. So we came up with a network of networks. So we take all those networks, the 21 networks, and then we compute the hardwired cluster distance between each of them. And then we throw that into NeighborNet, and then we get this nice picture here. If you look carefully, nearly everywhere there's exactly 2 data points. One is from the one algorithm, one is for the other. So we immediately see which pair from the 9 versus the 12 match up. There's only 3 locations where there's 3. So 9 plus 3 is 12. So that all works out. And so that gave us an easy way to figure out without having to spend hours trying to figure out, are they the same or are they not the same? And then we can use our tangogram viewer to then actually take 2 of the ones that shouldn't be the same, and then have a look at them and actually figure out what's going on. Then so we see here, if you see, look at this taxon, if you follow up to the root, there's actually 1, 2 reticular edges. So the reticular edges always drawn as little curves here, and over here, if you follow this up through, it's only got one. So that's where they differ. And then we actually figured out that the one algorithm is allowed to do this. It's allowed to have a node where there's two reticulate edges coming out. And so that's the multiplicating algorithm. And the other algorithm is not allowed to do that. It has to be, I think there's a technical name, normal or something like that, where each node has, no node's allowed to just have reticulate children. Well, it's allowed here, and that's why there's 3 extra in that one, because it's slightly more general algorithm. So that's the network of networks. And I don't know how many of you know that when we were here 2007, we came up with a book project. I was here with Regular, she was a postdoc in those days, and we started working on a book describing stuff in phylogenetic networks, and now 5 years later or 4 years later, whatever, it's here. So if you haven't had a chance to see it, I'm very happy to show it to you, and it's available in the bookstore. Okay, thank you.
We obviously have time for questions.
I have actually 2 questions. You were saying that in this hybridization network approach that you implemented, the new one, the minimal hybridization,
Yeah.
you're not using maps. What are you using?
We're using partially processed trees where each leaf can be dead or alive. Okay. I
have another question.
So I guess we're not thinking in terms of maths and stuff like that, but I'm sure that you'd be able to. So the basic idea is you start with 2 trees, and then you start killing off different leaves, but you leave everything there. It's not like you take stuff out. And then the nice thing about that is, so you keep killing leaves until suddenly the 2 trees, that's what's left over. What's left over, the alive parts are isomorphic, and then you can put them together. And then you don't have to worry about where do I attach my reticulation nodes, because they're still in there. You haven't taken them out, so you don't have to put them in. And so that makes it much easier than first breaking something down and then in some random order adding stuff back in. So it's a pretty pedestrian in a way, but it seems to work.
And the other question I had, you were comparing networks with networks. Now there are a number of metrics out there by now.
Yeah, that was just the hardwired clusters.
Yep.
That's
My question is, many of these metrics are actually only distances on networks. So how robust is yours? Because you could, for example, use Valiente's the M metric by Nacle.
Well, the reason, this is nice, isn't it? Because you've got 2 networks. You've got all these networks contain the same 2 trees, and yet you've got this wide spread of stuff. So that's kind of quite nice. So you wouldn't want to use the contained trees metric, because then they'd all be in one point, which would not be good.
But you could use, for example, the path metric that Valiente work with.
The reason why we're not using that is because we haven't implemented that yet.
Okay. No, we will.
So actually, Benjamin, who was just mentioned earlier, we're hoping to hire him for the summer to implement different things. Yeah, the nice thing about the hardware cluster is it's easy to compute. And for this particular thing, it worked fine.
Another question?
So for the ocean metagenomics data, you were saying that it's easier to find protein coding hits. So does that mean that there's much less signal in the non-coding regions? How do you access that?
Sorry, it was easy to find. When I said it was easy to find,
So you were saying that you were so you didn't want to do a DNA blast because the sequences weren't there, but you were doing blast X.
The reason why you do blast X and not a DNA blast is because when you compare on a protein level, you can see further back in time.
Right. No, but
see more distant homologies.
To what extent are you then losing out on the non-coding signal, even though it's
the sequences of that non-coding, obviously you're gonna lose those, and so you could try and do both. You could take the sequence. I mean, as data sets get bigger and bigger, people are going to have to do more and more of a multi-stage process. I mean, you probably will use a fast short read aligner to try and skim off the 1% that you can get with those, and they're already assigned, and then you might do a blast X or a blast N comparison, and maybe assign another certain chunk. And actually we did something on the side which is a bit worrying. If you take 16S sequences and blast it against the NR database, you'd assume no hits. But actually you get a nice spread of hits. You get something that looks like a real metagenome. And so whenever someone has DNA sequences, some of those sequences are going to be 16S, and they blast them against the NR, and they get all these nice hits, and they're just wrong. So there's a lot of bad stuff in the NR database. There's a lot of 16S sequences annotated as coding genes.
You mentioned the kind classification in a slide.
Yeah.
Can you say something about it?
The chime kind, whatever it's called. That was just, I don't know much about that. All I did was added an importer. So you can do whatever kind or chime does. And then when you finish, you get a file and you can just put it into Megan and Megan will display it for you. So it's just a way to try and make this program. So, the program, you can think of it as a glorified viewer. So you've got any kind of classification, the program, we hope that you can just put it into Megan and see it.
You have another question?
Yeah, it's about the usage of Blastix against those reference database.
Yeah.
Have you tried to quantify the number of fragments you're missing because of frame shift?
Yeah, I think that is a big problem because we've done simulation studies where we compared Illumina sequencing versus 454, and 454, has that problem that sometimes it misestimates the number of homopolymers, so you get frame shifts more easily. And in our simulations, at least, Illumina always work much better than 454 just because of that one feature. If we turn that feature off, then they work the same. It's just sequences. Yeah, so frame shift is a killer for matches.
Just another one. You just said that comparing with the protein level, allowed to compare the back in time.
Yeah, or more distant.
The similar idea would be maybe to use some profiles, phylogenetic, so just
that might be
hammer or something like this to look further back in time and then extend from your reference sequences to something that's never been sequenced yet.
Yeah, that would be an idea. We haven't tried that, but.
Okay, go ahead.
What do people propose to do with these short reads that are unassigned? What's the next step?
Unassigned. Well, for a start, there's different machine learning approaches where you're looking at different KMA counts and so on, and you're hoping that there's some correlation between the typical KMA profile of a particular organism and the reads that come from it. So even if it's not present in that in the reference database, you might still get lucky and see, okay, this just looks like E. coli, even though it doesn't match anywhere in particular. So that's one approach that people are using. Yeah.
I have two questions for you, which I think I'll sneak in before everyone else has more questions. They're related. You're inserting sequences into the NCBI taxonomy, but there's already been a fair amount of discord about the NCBI taxonomy, so that's basically one topic.
Yeah.
And the other one is there's two fundamental approaches to this kind of problem, and one is the kind that we're talking about here, which is looking at features about sequences, and the other one is like this taxonomic placement using sequence alignment, right? And measurement for the second type of thing, measurement error, like evaluating the accuracy there, has one way of doing it. Evaluating the accuracy of the first really is looking at supposed truth for the taxonomic identification. What's your comment about how to do that evaluation correctly? How do you know the correct species? How do you know that kind of information?
How do you evaluate how well the
yeah, I mean, you didn't show any accuracy measures here.
Yeah.
And so the question is, I think that's really a fundamental question. How to do that?
So we've done. So what people do is they take a database of. One way that you can evaluate this kind of method, you take a database of known sequences, you simulate the sequencing part, and then you map against the NCBI taxonomy. But of course, the problem there is that you're only modeling those sequences that we already know, and it's difficult to. There's the known unknowns and the unknown unknowns, but we don't know the unknowns. So we don't know how to simulate the sequencing from unknown sequence. So what people normally do there is say, okay, let's take out a whole subclade of the reference database and pretend that the stuff that we, like take out all the epsilon proteobacteria, for example, and then pretend that every sequence that comes from the epsilon proteobacteria in the simulation is coming from some unknown source. But
do you trust using the NCBI as a reference?
I know that, so Megan doesn't force you to use the NCBI taxonomy. You can supply your own tree in your own mapping file. And I know that a number of people do that. So there's some people who take the silver taxonomy and they modify it in Bergey's taxonomy. The way that, I mean, one way to think of it is, if you think of the taxonomy just as a way to present your results, rather than the main means of analysis, then it's fine. And then you're not making much of a mistake, I'd say. You know, I mean, you could separate the two out. You say, okay, I'm not going to use the NCBI taxonomy. Yeah, so yeah, let me say it this way. Megan allows you to import from a number of different analyses, and the common way, so you want to be able to compare different analyses, and the common backbone is going to be the NCBI taxonomy. So you do something based on the silver taxonomy, and then you import it into Megan, and Megan's going to a degree, reshuffle some of those results so it fits into the NCBI taxonomy. So I think it's very valuable to have one taxonomy to be able to compare things. And of course, there are problems with the NCBI taxonomy, but yeah, I don't know how to resolve those.
Other questions?
There are people who argue that it is unfair to make comparisons between two metagenomic samples without going through certain type of normalization between the two data sets. And I think you didn't talk about that. Yeah. Do you do some type of...
Yeah, so I mean, what's implemented in Megan at present is overly naive. So you've got some different data sets, one's very big and one's very small. Yeah. What we do, and we know we shouldn't do it, is we just multiply the numbers so they're all based on, say, 100,000 reads. And so the problem with that is if your data set's only got a very small number and you multiply all the counts by something, then you're underestimating the low abundance taxa, because if you've only got 10 taxa to begin with, you still have 10 taxa, but each one will have a large number of reads. And so the better way to normalize would be if you have a small data set and a big data set, you randomly subsample multiple times from the big data set to the small size, and then so that's something that's the way one should do it. It's not implemented at the moment just because it's painful to implement.
Any other questions?
I guess the biologists are thinking very much in terms of comparing samples which might be drawn from different locations.
Yeah.
And again, I guess that they're interested in making inference about what's going on. So I mean, to understand it, the sort of the origins of metagenomics were really in terms of thinking of rather than think of individual organisms which sort of may interact, but you just treat them as an ensemble of things which somehow represent a sort of super organism. And therefore, when you're comparing samples, you're trying to compare the kind of processes which are going on. And you did talk about this.
Yeah.
But I wondered how much is known. I mean, if you're trying to sort of map your reads onto particular genes in particular enzymatic pathways in KEGG or something, and you're trying to make inference about the processes, I just wonder, is it really legitimate to kind of imagine that, the bacterial cell was really, the wall wasn't really there.
No,
and
that's what I said earlier. That's some of the interesting stuff. So once you've got a KEGG picture where all these different enzymes light up, you're not done, because that's really based on the idea that there's one organism who's supplying the transcripts. But now we've got a mix of different organisms. And if I see a pathway where 50 different organisms are involved, then that's not real. That can't be real.
I was just wondering whether there's any kind of knowledge about which kind of processes could literally be done by different bacteria sort of exchanging material somehow, and which ones would really have to take place within the same cell. Is there a knowledge of this? Or is it
I don't know. So I mentioned briefly a mouse gut project that I'm involved in Tübingen, where they have the sterile mice, where they can determine what the gut metagenome is going to be, and with some guys who can do protein sequencing using immuno reaction. I don't know. They use some kind of protein sequence, and they can determine whether the protein comes from the one. Well, we're looking for proteins that are very similar across along the sequence, but differ at a certain place, and we'll be able to say, okay, whether it comes from the one and from the other. So we're trying to. That's one thing we'll try and do, is try and figure out that's not directly answering your question, but we're trying to figure out if we see a certain protein, who's supplying it. So that kind of thing. I don't know which kind of networks, which kind of pathways are the ones that one should look out for when it comes to bacteria coming together to make that possible. I don't know, maybe in biofilms, I mean, where a biofilm that involves more than one type of bacteria, maybe there's some, maybe even there they're not cooperating directly. Maybe some guys are doing something because they're on the outside and the other ones are in the inside and they're providing something differently. That might be the...
I guess it sort of depends whether or not the proteins are somehow excreted and somehow the bacteria sort of live in the soup that they create themselves and mutually benefit from the soup that they're all making together. But I don't know if this is what people think about.
Okay, so I think that we should thank the speaker. Thank you very much.